First of all, I would like to thank the organizing committee for selecting my abstract for as oral presentation in this international conference on applied zoology. My topic is very new for most of the audience, and I hope it will be interesting and will helpful for exploring the new research domains for most of the people. And this topic is basically multi-parameter detection of non-cell autonomous and cell autonomous death in cancer cells. Myself, Dr. Syed Qasim Raza, and working as assistant professor in IBBT UAS. First of all, we have to know about the types of cell death. There are basically, they are programmed cell death and there is accidental cell death. So type 1 and type 2 are programmed cell death like apoptosis and autophagy. Necrosis is type 3 and is accidental cell death. We classify it as an accidental cell death. There are some other deaths which have been classified recently, and those are like atypical cell death, like mitotic catastrophe and entosis. Basically, this is the entosis to who we called non-cell autonomous cell death. And when we called it the cell death as the programmed cell death, caspase dependent and caspase independent. Apoptosis is caspase dependent, and the other ones are like autophagic and necroptosis. When necrosis is programmed, then necroptosis is programmed cell death. So interestingly, this phenomena of cell-in-cell internalization or cellular cannibalism or entosis was first time studied by Overholzer and colleague in 2007, and they have said that when two cells are coming together so close to each other and they interact with each other, and after some hours, one cell is going to be internalized inside the other cells like a phagocytosis. So in this cell, there was one target cell, other was the host, and target was going to be invade inside the other cells, so host cells. But they confirmed their results by interestingly finding that these cells, which were going to be internalized through this look like phagocytosis phenomena, were not like phagocytosis, because these cells were the live cell, and when they co-culture green and red cell together of basically same cell lines, like MCF7 or MCF10 breast cancer cell lines. So they found that these cell-in-cell structures in their cultured cells. And after going for imaging, they found very interesting facts that these cells were just not overlapping outside. The red cell was not overlapping on the green cell, but it was completely internalized inside this green cell. So this complete internalization was confirmed by the confocal microscopic x-axis and y-axis images, and then was also confirmed by electron microscopy that there is this circled area, like seam cell is complete cell is inside the other cells, and this show that this is the nuclei of this host cells, and here is the nuclei of the nuclei of the guest cell, and here is the nuclei of the host cells. After knowing about this, they confirmed their results by using the ZVAD inhibitor, which is the inhibitor of apoptosis, BCL2. When you overexpress BCL2, it means you are going to overcome on the apoptosis, and combine together BCL2 and ZVAD. They found that there was no reduction in cell structures. So they confirmed their result that this process is independent of the apoptosis, and they can say that this is non-apoptotic cell death process. They also confirmed their results. The results were confirmed like cathepsin B substrate, lysotracker, LUMP1. LUMP1 is basically lysosomal associated membrane proteins. And they confirmed through beta-catenin, which was the marker of the cell boundary. So they found this big giant cell having this cellular structure inside by lysosomes accumulated like a ring-like structure. So they confirmed that this cell is completely internalized inside the cells, and that will die through the cell-in-cell internalization. And they also found that these red colored show that these cells are basically inside, and they are died through the lysotracker and cathepsin B staining positive, and they are dying through the lysosomes. So they say that the death of this after cell-in-cell internalization, so death is the lysosomal cell death. They also found some interesting facts regarding the blocking and cadherin antibodies. They found that there are the E-cadherins or P-cadherins. The cadherins are basically involved during these cell-cell interactions. And when you are going to block these cadherins, so you are going to inhibit the cell-and-cell internalization. So they use some chelating agent like EDTA and EGTA with some E-cadherins. So they found, interestingly, that there is the reduction of cell-and-cell internalization. Also the same for the MCF10A cell lines. After using these E-cadherin and P-cadherin antibodies, they found that after 12 hours in control, they have the 30% cell and cell internalization, but after 12 hours when you're using the E-cadherin, so these results confirm that there is involvement of cadherin at the junction of the cell and cell internalization. The interesting thing other regarding this entosis phenomena was this, that the cell was sometime going to be released after internalization like this. This was cell inside, and after some time the cell was released, and after releasing the cell can divide and live happily. The same for the other cell, that when cell are internalized inside the other cell like this, these cell inside the cell can divide. So this was also a very interesting phenomena, that so when there are now, you can say there are three ways. One is the cell is going to be die inside the other cell, other is the cell is going to divide inside the other cell, and third one is cell is going to be released from the other cell. These results confirm that the cell which are internalized are healthy cells. So they also confirm these studies in human tumors. They stain the human tissues with different markers of their cellular boundaries and their nuclear staining. They found like this structure, if you see, this is the complete cell boundaries, and this is the complete cell boundaries of the internalized cell. This show that the cell which is inside is completely live cell, is not like debris or something else. For us, we did our results also using these facts, because at that time this mechanism was not understood what will happen to the host and internalized cells. So we also did this study, cell and cell internalization. We also found cell and cell structure and different cell lines. We found that most of the cancer cell line have these cell and cell structures. And we also confirmed our results by these XZ and YZ and YZ imaging. So different axes confirm that these cells are not just like floating cell, but they are just cell which are completely internalized inside the other cells. So just a little video that you can see from my study, that how the cells are going to be internalized inside the other cells. So this green cell will be internalized inside the red cell. The same population is the same, but we only colored them through different cellular markers. After knowing about this, so for us, there was issue that the cell were going to be sticky together. Cell were just sticky cells. By using the flow cytometry, we wanted to know, isolate the double positive cells, but there was the issue that cell which are sticky will also be double positive, and they can be isolated from the flow cytometry as a double positive cells. So this problem was solved by our laboratories there, and they have found one technique which was image stream flow cytometry. So in image stream flow cytometry was just new technique at that times, and we found very interesting result during this by using this technology. So for example, you have the cell death type 1 and type 2 apoptotic, mitotic, pyroptosis, and autophagic by using an Axine 5 and REC7 staining like markers, and the same for the flow cytometry. For us, flow cytometry, for example, was a little bit different from the flow cytometry, because in flow cytometry, we have the passage of the cell like double positive, single positive, but at the same time, there was the laser light which was putting on the cells like this, and we get the image of these cells which are going to pass. So you have the passage of the cell, and at the same time, you have the imager which is going to take the images of these cells. So you can confirm your results that either these double positive cells, for example, here, if you see here different channels, green channel, red channel, and then the blue channel, and like the orange channel, and far red channel, and all are going to be submerged. And if you see here, this is green cell which is going to be internalized inside the red cell, the same double positive cell that we called it non-cell autonomous death. And these cells show double positive, and this by flow cytometry, and also were confirmed by these cells were confirmed, that the cell were completely internalized inside the other. We did also some experiment on irradiation, so by using the radiated cell, and we found that the radiated cell were going to engulf the cell which were not irradiated, and we found their population in our experimentation. So due to we also did some experimentation on some inhibitor, and we confirmed that our cell and cell study was the same as like previously, that the cell death was non-apoptotic, and target cell percentage was cell which was internalized was degraded through the lysosomal cell death. So this image stream flow cytometry have multiple applications like cell signaling. You can study internalization, surface intracellular colocalization, DNA damage and repair, microbiology, cell cycle, cell death and autophagy, shape, stem cell biology, parasitology, cell-cell interactions. So you can have the flowing of the cell, you can have different color of those cells through flow cytometry, and at the same time you have the imaging of these cells. So thank you very much for your attention, and thanks for again I would like to thank all the people who are listening to my lecture, and thank you very much.